Hello everyone, good evening. Two minutes, let us wait and I will start the session. Okay, I got three worksheets. I guess, let me check here. Jyoti sent it and one minute. Yeah, Priyanka sent it and Anvita sent it. Priyanka and Jyoti's worksheet I already corrected and sent. Anvita's I have to correct and I will send it soon by the end of the day. Yeah, thank you guys. I appreciate your consistent performance in submission of worksheet. And I assure you, you will get success if you practice and if you maintain consistency. ಹಾಗೆ ಹೇಳ್ತಿದ್ದೀನಿ ಅಂತ ಈಗೇನು ಹೇಳ್ತಾರೆ ಮೇಡಮ್ ಅಂತ ಅನ್ಕೋಬೇಡಿ. ಎಕ್ಸಾಮ್ಸ್ ಆದ್ಮೇಲೆ ವೆನ್ ಯು ಗೆಟ್ ದ ರಿಸಲ್ಟ್ಸ್ ಯು ವಿಲ್ ಗೆಟ್ ಟು ನೋ. Okay, let us start. So in the last session, we discussed regarding the female reproductive system. ಅದಾದ್ಮೇಲೆ spermatogenesis ಬಗ್ಗೆ, sperm formation ಹೇಗಾಗತ್ತೆ ಅದ್ರ ಬಗ್ಗೆ ಕಲ್ತಿದ್ವಿ. In today's session, we are going to discuss regarding oogenesis, ova or egg production ಏನಾಗತ್ತೆ ಅಂತ. Before that, let us solve some questions on spermatogenesis. So be ready with your chat box, be interactive, ask the questions if you have any doubt. Okay, okay, fine. So let me proceed. Be ready with your books to scribble down the questions. Otherwise, I will be sending it. Neat. Okay. Just a minute. The screen is loading still. Yeah. Yes. So regarding the gametogenesis, the first question, I think the screen is visible to everyone. Is it visible, guys?
Yes,
Okay, fine. First question, meiotic division of secondary oocyte is completed in. I did not complete this, I guess. So just try or guess the answer because it is oogenesis there. Prior to ovulation, at the time of copulation, after zygote formation, at the time of fusion of a sperm with an ovum. Meiotic division of secondary oocyte is completed in. One ನಿಮಿಷ. Yes, I got the first answer from Jyoti. At the time of copulation, she said. Shri Suresh, okay. Option E, we don't have an option E here. Is it visible? Option E is visible, Shri Suresh. Nagini option D, Manikantha option, very good, Q. Nagini B option D, Shri Suresh option D, Manikantha option A. So the right answer is prior to ovulation only. So let me explain you later. ಯಾಕೆಂದ್ರೆ ಇದು oogenesis ಇದೆ. This is related to spermatogenesis. You can try. The difference between spermiogenesis and spermiation. Difference between spermiogenesis and spermiation. Option A, in spermiogenesis, spermatids are formed, while in spermiation, spermatozoa is formed. Option B, in spermiogenesis, spermatozoa are formed, while spermiation, spermatids are formed. Option C, in spermiogenesis, spermatozoa from Sertoli cells are released into the cavity of seminiferous tubules, while in spermiation, spermatozoa are formed. D, in spermiogenesis, spermatozoa are formed. Just a minute. In spermiogenesis, spermatozoa are formed, while in spermiation, spermatozoa are released from Sertoli cells into the cavity of seminiferous tubules. Yes, guys, what is your response? Okay, I got the first quick answer from Netravati. Okay. What about others? Jyoti, Priyanka, Manikantha. Option A, Priyanka and Jyoti voted for option C. Please select to personal chat, because you have to send your answer to the personal chat to Neat Sankalp. Okay, many of you are sending to everyone, so please change that option there. Yes, what about others? Many are there, no? 9 students are there here. Sorry, 8 students are there. Anvita, Nagalambika, Shri Suresh, Nagini. What is your response? Whether the questions are visible or no? Option C is the right answer. Which of the following layers in antral follicle? Idu beda because navinnu madilla adana. Which of the following cells during gametogenesis? Yes, Shri Suresh, any doubts? Okay. Which of the following cells during gametogenesis is normally diploid? Yaudu diploid alirate? Spermatogonia, secondary polar body, primary polar body, spermatid. Which will be diploid? Yes, guys. Jyoti answered it. Okay. What about others? Gametogenesis time alli. Manikantha answered it. Priyanka answered it. Okay. What about others? Nagalambika, Anvita, Nagini answered it. Option B. ಯಾಕೆ ಆನ್ಸರ್ ಮಾಡ್ತಿಲ್ಲ ಯಾರು? Option B, secondary polar body. Nagalambika, option B. Fine. So let me explain you first, then I will ask these questions. Please be interactive. ತುಂಬಾ ಜನ ಸೆಷನ್ ನಲ್ಲಿ ಇದ್ದೀರಾ, ಆನ್ಸರ್ ಮಾಡ್ತಿಲ್ಲ. What is the reason? It is not a movie or a serial which you are watching. Be interactive. You all are living organisms and you are attending biology session, right? Okay. So regarding the spermatogenesis, we have another concept which is called as hormonal balance. So just let me know whether we completed spermatogenesis completely. Yes? Okay, fine. So hormonal control, this spermatogenesis is there, no? This is under the hormonal control. So what happens actually? Did I complete this or no? I think I did not complete this. Hormonal method agidena, hormonal control of spermatogenesis completed. Okay, thank you. Okay, thank you. Next, we will go to the structure of a sperm. So as you can see, the structure of a sperm in this image, we have the parts like acrosome, nucleus, neck, mitochondrial region, and these three main parts, head, middle piece, and tail. So let us study what is there. Ultrastructure of sperm consists of head, neck, middle piece, and tail. Then the sperm head contain an elongated haploid nucleus. You can see the nucleus here. It is elongated and haploid, single set of chromosome. Ploid is haploid. Above the nucleus, there is a cap-like structure. You can see this cap-like structure, which is called acrosome. ಅದಕ್ಕೆ acrosome ಅಂತ ಕರೀತಾರೆ. The acrosome contains enzymes which help in fertilization of the ovum. So acrosome is very important because it will be having the enzymes, it will be having the chemicals which help in or help during fertilization. Next, the middle piece contains, idu middle piece alva. So the middle piece contains mitochondria. We know that mitochondria are called as powerhouse of a cell. So what it does, it provides energy for the movement of a tail. This is a tail. So it provides energy for the tail to move and support sperm motility or sperm movement. Next, human male ejaculates 200 to 300 million sperms during coitus. That is, during the sexual intercourse. One sexual intercourse, there will be a release of 200 to 300 million sperms. Then 60% must have a normal shape and size. 40% of them must show vigorous motility. So our 200 to 300 million sperms alli 60% they will be having normal shape and size irutte. Uldid 40% alli they will be showing very active motility or the movement. So sperm released from the seminiferous tubules enter into accessory ducts. Avenagtave accessory ducts ige hogtave. What is that? This one, vasa difference and all, accessory ducts ige. Male reproductive system alliro accessory ducts ige hogtave. Then after that, on their way, they will get the secretion from the accessory glands. Along with that, the seminal plasma, the sperms become semen. So semen enagatte, it will be released during sexual intercourse. So the function of male accessory ducts and glands are maintained by testicular hormone androgen. So ಇದರ function ಇರತ್ತಲ್ಲ, accessory ducts ಗಳಾಗಿರಬಹುದು, accessory glands ಗಳಾಗಿರಬಹುದು, ಇದರ function ಅನ್ನ ಯಾರು ನೋಡ್ಕೋತಾರೆ? ಆಂಡ್ರೋಜನ್ ಹಾರ್ಮೋನ್ ಅಥವಾ ಟೆಸ್ಟೋಸ್ಟಿರಾನ್ ಹಾರ್ಮೋನು ಇದರ ಫಂಕ್ಷನ್ ಅನ್ನ ನೋಡ್ಕೊಳ್ಳುತ್ತೆ. So this is regarding the structure of a sperm. Now we have the other process which is called as oogenesis anta. So this is a oogenesis, right? So ಇಲ್ಲಿ fetal life ಅಂತ ಇದೆ. Fetal life ಅಂದ್ರೆ ಏನು? The oogenesis occurs when that baby girl, for example, I have a daughter, okay? My daughter's oogenesis occurred when she was in my womb. She was in my womb, okay? So the oogonia is present in the fetus. Oogonia anta karitare. So that oogonia enagatte, this oogonia undergoes mitotic division. Idenagatte oogonia annodu mitotic division agatte. Then it forms primary oocyte. Primary oocyte anna form ಮಾಡುತ್ತೆ. So this primary oocyte will undergo meiosis. Primary oocyte ಏನಾಗುತ್ತೆ? It will undergo meiosis. So in meiosis, we know that we have meiosis 1 and meiosis 2. So the first primary oocyte will undergo meiosis 1, completed prior to ovulation. So meiosis 1 ಏನಾಗಿರುತ್ತೆ ಕಂಪ್ಲೀಟ್ ಆಗಿರುತ್ತೆ ಓವ್ಯುಲೇಷನ್ ಆಗೋಕ್ಕಿಂತ ಮುಂಚೆ. ಸೊ ಮಿಯೋಸಿಸ್ 1 ಪ್ರೊಡಕ್ಟ್ಸ್ ಏನಿರ್ತವೆ ಇಲ್ಲಿ ಸೆಕೆಂಡರಿ oocyte and first polar body. So secondary oocyte in a haploid condition, primary oocyte is in a diploid condition. So this is first polar body is formed. So secondary oocyte will be at a what we can say resting phase. Secondary oocyte resting phase alla irutte. Then adult reproductive life bandaga, or you can say when the first menstrual cycle begins in a girl, or when she attains maturity, this secondary oocyte is there, no, it will complete its meiosis 2. Avaga en madutte adu meiosis 2 na complete madutte. So when it completes its meiosis 2, one ovum or a egg is released along with the second polar body. So these polar bodies there, no, these will degenerate, these will die off or wash off later. So this is regarding the oogenesis. So illi you have a video regarding that, please watch it carefully. And this is whatever you are seeing, no, this is a oogenesis, that is the section. The first one is zona pellucida, vitelline membrane, germinal vesicle, okay, nucleus, this is a ooplasm, and this is corona radiata. This is a structure of ova or a ovum. Okay, next. Okay, just a minute. Watch this carefully. Okay. Oogenesis. This is a female reproductive part, ovary, which produces the egg. So the process is called as oogenesis, production of egg from the ovary. Okay, this is the ovary, what you are seeing. In the ovary, you can find many follicles. See how beautiful they are. Many egg follicles are there, right? So the follicles are in different stages. Idu enu primordial follicle. Here you can see early antral follicle. Here you can see the matured Graafian follicle. Antrum ide, oocyte ide. This is a nucleus, right? Let us see what happens later. So the division occurs here. Idu follicles alli, whatever you are seeing, no, the division occurs in the nucleus, in a primary oocyte he is showing now. Primary oocyte diploid in condition alli irutte alva. So ivella ಏನು primary oocytes. This is still developing. Now what happens in primary oocyte? This primary oocyte which is present in a fetus, it will undergo meiosis. See, oogonium, right? So this oogonium, what it does, it will undergo mitotic division first. So from the mitotic division, oogonium. So here you can see the growth, the enlargement of a cell. The chromosomes you are seeing here, this is a primary oocyte which is diploid in condition. So meiosis 1 occurs, which is called reduction division. Here you can see the chromosomes are separating, right? So meiosis 1 occurred. So haploid polar body, first polar body is formed. And what is this form? No, this is a secondary oocyte. This is a secondary oocyte. So a secondary oocyte enagatte, it will undergo meiosis 2 when it comes to adult maturity. If when a girl's mature, it will undergo meiosis 2. Here you can see that. Okay, just a minute. Yeah, meiosis 1. Secondary oocyte, meiosis 2. So this is a first polar body, second polar body. This is a oocyte or a ova, whatever, oogonia, where we can say, no, oocyte. This is a ova or a egg. So these all are the polar bodies which will degenerate. So this is called as oogenesis. What will happen to this? This will be having the haploid egg or a oocyte, what we are calling. Okay. So higiddaga, once this form is reached, then when the sperm enters, the fertilization will occur. If the sperm will not enter, so the menstruation cycle happens and the period occurs where this ova or a oocyte will be, this ovum will washed off through the lining of the blood as well as mucus in the form of a blood. Okay. This is regarding the oogenesis. Artha aita oogenesis ಬಗ್ಗೆ ಎಲ್ಲರಿಗೂ? Yes, guys? Is it clear? Okay. Now I will be sharing the questions. I request everyone should interact in the session through the chat box or through the mic. Okay. ಈಗ questions ಅನ್ನ ಕೇಳ್ತೀನಿ because ಅವಾಗ ನಿಮಗೆ you got a questions from oogenesis, so you are in a little confusion. Now I will ask the questions. One ನಿಮಿಷ. ಎಲ್ಲಿತ್ತು? Okay. So third question ಇದೆ ನೋಡಿ ಇಲ್ಲಿ. Which of the following layers in an antral follicle is acellular? ಅಂದ್ರೆ cell ಇರೋದಿಲ್ಲ ಅಂತ. Acellular. It doesn't have any cells inside that. That is nothing but a stroma of fluid. Next, fourth one answer ಮಾಡಿದ್ವಿ. Fifth one, what is the correct sequence of sperm formation? Read it carefully, guys. Sperm formation sequence ಅನ್ನ ಹೇಳ್ಬೇಕು. Options are here. Spermatogonia, spermatozoa, spermatocytes, spermatids, spermatogonia, spermatocytes, spermatids, spermatozoa, spermatids, spermatocytes, spermatogonia, spermatozoa, spermatogonia, spermatocytes, spermatozoa, spermatids. What is your response, guys? Be fast. Sperm formation ಹೇಗೆ ಆಗತ್ತೆ? ಮಣಿಕಂಠ answered it. Good. Priyanka, good. What about others? Anvita, good. Nagini, option D. Okay. Yes, what about others? Netravati, okay. Fine. Guys, the right answer is here. Option, okay, I got the answer from Jyoti and Nagini. Okay. Option B anta. The right answer is spermatogonia. After that, spermatocytes, spermatids, spermatozoa. Option B is a right answer. Option B. Next, the figure shows a section of human ovary. Select the option which gives correct identification of either A or B. What they are asking, the correct identification of either A or B with function or characteristics. So A ಅಥವಾ B ಎರಡರಲ್ಲಿ ಯಾವುದು ಆಪ್ಷನ್ ನಲ್ಲಿ part is also correct, function is also correct, they are asking. So option A, B is a corpus luteum, it secretes progesterone. Option B, until I complete the question, please don't answer. Option B, A is a tertiary follicle, forms Graafian follicle. Option C, B is a corpus luteum, secretes estrogen. Next, option A, primary oocyte. A is a primary oocyte. It is in the prophase of meiotic division. What is your response, guys? Okay. Anvita answered it. Manikantha answered it. Nagalambika, Nagini, good. So option A, that is corpus luteum, which secretes progesterone, is a right answer. So option A is a right answer here. Option A. Next question. Just a minute. which one of the following, I repeat, which one of the following statement is false in respect of viability of mammalian sperm? ಯಾವ ಸ್ಟೇಟ್ಮೆಂಟ್ ಫಾಲ್ಸ್ ಆಗಿದೆ ರಿಲೇಟೆಡ್ ಟು ದ ವಯಬಿಲಿಟಿ ಆಫ್ ಮ್ಯಾಮೇಲಿಯನ್ ಸ್ಪರ್ಮ್? ವಯಬಿಲಿಟಿ ಅಂದ್ರೆ ಎಕ್ಸಿಸ್ಟೆನ್ಸ್ ಆರ್ ದ ಮಾರ್ಟಾಲಿಟಿ ಅಂತ ಏನ್ ಹೇಳ್ತೀವಿ. ಸ್ಪರ್ಮ್ಸ್ ಈಸ್ ವಯೇಬಲ್ ಫಾರ್ ಅಂದ್ರೆ ಹೌ ಲಾಂಗ್ ದ ಸ್ಪರ್ಮ್ಸ್ ವಿಲ್ ಬಿ ಆಕ್ಟಿವ್ ಆರ್ ಅಲೈವ್. Sperm is viable for only up to 24 hours. Survival of sperm depends on pH of the medium and is more active in alkaline medium. Viability of sperm is determined by its motility. Sperms must be concentrated in a thick suspension. What is your response? False statement keltidare makkale avaru. They are asking false statement. Okay, I got the first quick answer from Jyoti. Okay. Manikantha, okay. Priyanka, fine. They are asking false statement. Nagini, Anvita. So the right answer is here option D. Sperms must be concentrated in a thick suspension. That is a false statement, wrong statement adu. The sperm is viable for only up to 24 hours, right thing. Survival of sperm depends on pH, right statement. Viability of sperm determined by its motility, right statement. All these are three correct statements. The D statement is wrong. So option D is a right answer here. Sorry. Option D is a right answer and it is correctly answered by Anvita. Okay, option D. Eighth question. Which one of the following statement about human sperm is correct? Correct agide anta. Option A. Acrosome has a conical pointed structure used for piercing and penetrating the egg. Option B. The sperm lysins in the acrosome dissolve egg envelope facilitating fertilization. Option C. Acrosome serves as a sensory structure leading the sperm towards the ovum. Option D. Acrosomes serves no particular function. What is your response? Acrosome ಬಗ್ಗೆ ಇದೆ ಇದು. Correct statement they are asking. Okay, I got first two responses by Anvita and Priyanka. Yes, Jyoti, Nagini. What about others? Option A is a right answer. The correct sequence of spermatogenetic stages leading to the, I repeat, the correct sequence of spermatogenetic stages leading to the formation of sperms in a mature human testis is. Spermatogenetic genesis kaltidivi alva, so other stages anna keltidare. First one, spermatogonia, spermatocytes, spermatid, sperms. Second option, spermatid, spermatocytes, spermatogonia, sperms. Third option, spermatogonia, spermatid, spermatocyte, sperms. Fourth option, spermatocytes, spermatogonia, spermatid, sperms. Okay, Nagini, Anvita answered it. Who is that? Priyanka, na? Okay. Okay, Priyanka, Manikantha, Anvita. So option A is a right answer where spermatogonia or sperm mother cells, which become spermatocytes, which become spermatid, then sperms. Next question. 10th one. In humans, at the end of first meiotic division, the male germ cell differentiate into first meiotic division ಆದ್ಮೇಲೆ ಎದರಲ್ಲಿ ಮೇಲ್ಸ್ ಅಲ್ಲಿ spermatids, spermatogonia, primary spermatocytes, secondary spermatocytes. The first meiotic division, the germ cells or the sex cells will divide or differentiate into what? Yes, I got the first quick answer from Jyoti, Anvita, Priyanka, Manikantha, Netravati, Nagalambika. Secondary spermatocytes ಅಂತ ಹೇಳ್ತಿದ್ದೀರಾ, ಕೆಲವೊಬ್ರು ಪ್ರೈಮರಿ ಸ್ಪರ್ಮ್ಯಾಟೋಸೈಟ್ಸ್ ಅಂತ ಹೇಳ್ತಿದ್ದಾರೆ. ದೇ ಆಸ್ಕ್ಡ್ ಅಟ್ ದ ಎಂಡ್ ಆಫ್ ಫಸ್ಟ್ ಮಿಯೋಟಿಕ್ ಡಿವಿಷನ್, ಎಂಡ್ ಆಫ್ ಫಸ್ಟ್ ಮಿಯೋಟಿಕ್ ಡಿವಿಷನ್. ಸೊ ಸಮ್ ಆಫ್ ಯು ಹ್ಯಾವ್ ಅ ಕನ್ಫ್ಯೂಷನ್. Let me explain you. Yeah. See here, spermatogonia or a sperm mother cell which undergoes mitosis, primary spermatocytes is formed. So after the end of first meiotic division, secondary spermatocyte is formed. After the end of second meiotic division, spermatid is formed. So the guys who answered as option D, that is spermatocytes, secondary spermatocytes anta. So those people answered it correctly. Okay na? So option D, secondary spermatocytes, is a right answer, guys. Next, Sertoli cells are regulated by the pituitary hormone known as luteinizing hormone, follicle stimulating hormone, GH growth hormone, prolactin. Sertoli cells produces testosterone. So this production is regulated by which hormone? Anvita answered it, Manikantha answered it. Okay. What about others? Be fast. So the right answer is here option A, luteinizing hormone. Option A, luteinizing hormone. The middle piece of sperm contains option A, proteins, option B, mitochondria, option C, centriole, option D, nucleus. Protein, mitochondria, centriole, nucleus. What is your response, guys? Yes, I got 4 quick responses. Jyoti, Manikantha, Priyanka, then Netravati, Anvita. Okay, good. So all of you voted as option B, mitochondria. It is a right answer. Next, how many sperms are formed from a secondary spermatocytes? They are asking secondary spermatocytes in the how many sperms are formed? Option A, 4, option B, 8, option C, 2, option D, 1. The first answer is given by Anvita as option A. Then Jyoti, Netravati, Nagini as option A, as well as Priyanka, Nagalambika, Manikantha. 4 anta heltidira. How many sperms anta? So let us confirm it. So in this diagram, you can see secondary spermatocytes ಏನಾಯ್ತು? Secondary spermatocyte will undergo meiotic division, right? So meiotic division ಆದ್ಮೇಲೆ ಒಂದು secondary spermatocyte will undergo meiotic division and forms spermatid. So these 2 spermatids are formed. These 2 spermatids develops into sperms. So ಒಂದು secondary spermatocyte ಇಂದ meiotic division ಆಗಿ ಎಷ್ಟು spermatids ಗಳು ಬಂದ್ವಲ್ಲಿ? Anyone now? How many spermatids you got from a secondary spermatocytes? 2 spermatids ಬಂತು ಅಲ್ವಾ? So 2 spermatids spermatids in the, we got 2 sperms. So the question is what? What is the question here? Confuse ಮಾಡಕ್ಕೆ ಇರ್ತಾರ ಅವರು. So ಇಲ್ಲಿ ನೀವು question ನೋಡಿ ಮಕ್ಕಳೇ. The question is, how many sperms are formed from a secondary spermatocyte? ಒಂದು spermatocyte, secondary spermatocyte ಇಂದ ಎಷ್ಟು sperms ಗಳು ಬಂದವು? Option C, 2 is a right answer. Option C, 2 is a right answer. Next question. Egg is liberated from ovary in. Egg is liberated from ovary in what? So the answer is, sorry, options are secondary oocyte stage, primary oocyte stage, oogonial stage, mature ovum stage. So it actually fertilization al baratte. Egg is liberated from ovary in which stage? Secondary oocyte stage, primary oocyte stage, oogonial stage, mature ovum stage. Graphian follicle kaltagu nan helidde. Priyanka said option D. Okay. Jyoti option A. Nagalambika, Nagini answered it. What about others? Anvita, Manikanta. Okay. So let me explain you that. So this we will learn in a Graphian follicle, that is female reproductive system. Yeah. Primordial follicle, primary follicle, then mature Graphican follicle, ruptured follicle. This is a liberated ovum. This liberated ovum itself a secondary oocyte, guys. Secondary oocyte. So you'll know it. A mature follicle, which is large fluid filled follicle that is ready to rupture, expel its secondary oocyte. Expel its secondary oocyte. So options ಅಲ್ಲಿ ಏನೇನಿದೆ? Mature follicle ಅಂತ ಇದೆನಾ? Let me see. Secondary oocyte. Question no. Egg is liberated from ovary. Ovary in the ಯಾವ stage ಅಲ್ಲಿ? Secondary oocyte stage ಅಲ್ಲಿ mature follicle in the release ಆಗತ್ತೆ. So the right answer is option A, secondary oocyte stage. Okay. So this is about few questions on oogenesis and spermatogenesis. So in the next session, we are dealing with fertilization concept. Sorry, menstruation cycle. ಅದಾದ್ಮೇಲೆ fertilization concept. ಇದ್ರಲ್ಲೇ ಯಾಕೆ ತಗೊಳ್ಳಿಲ್ಲ ಅಂತಂದ್ರೆ ಈ ಸೆಷನ್ ಅಲ್ಲೇ I approximately need 20 to 25 minutes for the menstruation cycle, and for fertilization event I need around 20 minutes for explanation. Along with the video, again it takes a lot of time. So I can't start in this session at 6:45. Only 15 minutes is remaining. I can't start both the concepts. So this is about the gametogenesis question. I hope the concepts are clear for you all. Yes? Concepts clear agidena? Everyone? Any doubts? Okay. So let me wind up the session here. Meet you all soon on Tuesday. Until that, take care. Have a nice day. Bye-bye.